Okay, hello everyone and welcome to our April WMISA seminar. Before we get started, I'd like to acknowledge the people of the Kulin Nation on whose land I am today. I pay my respects to their elders, past, present and emerging. Indigenous Australians are the oldest continuous culture and have studied and understood the Australian land and environment for more than 50,000 years. They were the first Australian earth and environmental scientists. The WMISA seminar series is about showcasing the amazing work of women in earth and environmental science in Australasia. As well as the live seminars, we've also collated recordings from seminars that are hosted by other organisations, and we put them up online on the WMISA website as well as on our YouTube channel, so you can check those out there. The live series is on the first Wednesday of every month, and the next one will be on May 5. You can sign up as a member of WMISA to get a notification of the upcoming seminars, or you can follow us on Twitter. Okay, so as you're no doubt aware, today's seminar will be presented by Dr. Indrani Mukherjee, who is a postdoctoral researcher in geochemistry at Codes at the University of Tasmania. She completed her PhD in 2018 from the University of Tasmania, and her main focus has been on understanding pyrite, trace element and sulphur isotope geochemistry in Precambrian marine black shales. Her research ties past geochemical conditions of the atmosphere-ocean system to the evolution of early complex life and the secular distribution of ore deposits through time. She aims to apply the pyrite LA-ICP-MS technique towards developing a deep time model for evolution of complex life and devising vectors to SEDEX zinc lead and sedimentary copper mineralisation. Well, that sounds fascinating, Indrani. So without further delay, I will hand over to you.
Thank you, Melanie, for that wonderful introduction. I'll begin with, give me 1 second, thanking WMISA and particularly you, Melanie, for inviting me to this talk today. And I'll be talking to you about the brilliant billion time in Earth's history. But before I go straight into my research, I'd like to share a few PhD related things with you because I found that was very important for me, my PhD, and it has really shaped my research in my career so far. And I just thought of sharing a few key highlights of my PhD and how that's shaped my research and encouraged me for my academic pursuits. If you can hear a noise in the background, it's because I've got a Formula One race after this talk, so I'll be jumping into that. Apologies for the sound. Yeah, so getting a PhD is probably not as important or remarkable as landing on the moon, but it can be very remarkable for someone in their career. Since I've mentioned landing on the moon, I might as well compare it with landing on the moon. So how does getting a PhD compare with landing on the moon? It's obviously a very small step for a man, everyone's heard that saying, but in case of a PhD, it's a giant leap for one person. And it's a giant leap for mankind in terms of landing on the moon, but it's probably a tiny, insignificant step for mankind when it comes to your PhD, unless you're Stephen Hawking, of course. And so today's talk will be about my tiny contribution and how that's encouraged me to pursue a career in academia so far. And I have, as I'm sure most of you have done your PhD or currently doing your PhD know, it's a very stressful event in your life, but it also gives you a fantastic opportunity to understand, see for yourself what interests you the most and what doesn't. And I've had a fantastic time during my PhD, and it's really opened up a lot of my research that I do now has basically stemmed out of my PhD. So I'll give you a bit of a background and then focus on two main aspects of my research today, which is the fundamental and applied using pyrite chemistry, some key findings that may be of interest to our audience here, and that will be followed by conclusions. Just before I thank everybody in my team, as we all do before talking about our research, I'll share this slide with you. This is a small piece of paper that my PhD supervisor wrote very quickly, 5 minutes when I first started my PhD. Oh, step 1, 2, 3, that's what you've got to do. And at the time, I was like, oh, that looks easy. And a phrase that he used quite often was take it easy. If I were you, I'd double check with your PhD supervisor if they actually meant it, because mine didn't. So I think I understood that early on. He says it, he probably doesn't mean it. He probably wants me to work hard. And so he said that a lot though, and I still have that piece of paper. Just reminds me of how simple things can look in the beginning and ordered, but it's the 3 years that follows from that time is a lot more complex, of course, but I really still hang on to this piece of paper because of fond memories. So here's Prof. Speaking of Prof, Professor Ross Large was my PhD supervisor. Even today, I still work very closely with him, and here's my team. Also, I'd like to clarify, this is not my twin sister with exactly the same name. It's one of those things where you go, I've got to change that, and just you never do. And so yet again, I will end up thanking myself with the rest of the team here. So 2 major research aspects, and that has been utilizing pyrite chemistry for mineral exploration. And so far I have focused on zinc-lead SEDEX deposits and sedimentary copper deposits. But the other aspect, which is very close to my heart, is applying pyrite chemistry towards understanding ocean atmosphere redox evolution. What can we learn about past atmospheric oxygenation cycles and ocean oxygenation cycles, including nutrient content and their fluctuation through time, and how that may have affected evolution of life in general. And that is absolutely very fascinating to me. And pyrite chemistry, why? Because, well, I've got the pyrite disease, and it's called being obsessed with the mineral. And that's because it's incredibly versatile. You can, as I just mentioned, you can understand ocean chemistry through time. Conventionally, people have used bulk rock, black shale chemistry, or other lithologies, but we focus on the single mineral, and not just ocean chemistry, but you can also gain insights regarding atmospheric oxygenation through time. I'm sure most of you know how useful pyrite, and particularly different generations of pyrite, is when it comes to ore deposit studies, as it highlights the fluid composition of the various hydrothermal pulses that have been there in an ore deposit system. And evolution of life as well, not just evolution. The other aspect of life is origins. Historically speaking, pyrite has been very subject of sort of strong interest from researchers that work on origins of life, because they find the structure of the iron monosulfide and disulfide to be very interesting in terms that it concentrates organic molecules and orders or sort of structures their layering to form the first cell membrane. So there's lots of information out there that will tell you why one can be, such as myself, obsessed with the mineral. And I also show you here, I'll just get the pointer for you. Yeah, so I've got the pyritized stromatolites here from the Dresser Formation, which is probably the first preserved evidence of life, first life on Earth. So it's an incredible mineral in terms of the opportunities or platforms it allows you to venture into, to find different things, different strands of research lines that I've been involved with, and I'm sure others are too. And when you combine that with advent of technology, it just becomes a huge database for information. And that's something that our team has been focusing here at CODES. But really it started in 2007 at CODES, which is the Center of Ore Deposit and Earth Sciences. Back then it was the Center of Excellence in Ore Deposits. And really my supervisor, Professor Ross Large, I call him Prof, And really, Prof and his team at the time used the chemistry of pyrite, combined it with laser ICP-MS. It was a rapid utilization of that analytical technique. It's very common now, but back then it was quite, it was breakthrough technology, really. And they utilized the information that they got out of these various different generations of ore pyrites, or ore deposit related pyrites, to gain information on the various pulses of hydrothermal fluids, etc., etc. But really, it clicked, and it was more like a eureka moment for my supervisor, thinking, well, if hydrothermal pyrites can provide insights into orogenesis, why can't we use sedimentary pyrite that forms in the marine environment to track its chemistry or changes in the water column chemistry through time? And that's how the project started. And so far we have about 10,000 sedimentary pyrite analyses in our database, which is just phenomenal, because you can use it for various different things. And I'll show you how we're using that, some of it at least. So what is the premise of using sedimentary pyrite in marine black shales? What we do is, the underlying premise is that oxidative weathering, and it has to be oxidative weathering because of the various redox reactions that are possible because of availability of oxygen in the atmosphere. So oxidative weathering, basically combined with erosion, is the dominant source of some of the nutrient trace elements into the oceans. So a combination of oxidative weathering, which is breakdown of rocks and materials, and transport of that material via riverine flux into the ocean, is how most of our nutrients get into the biotic realm in the oceans. And once they make their way into the oceans, they are utilized by the various organisms that thrive in the water column. And when they die, they become part of the mud, and it is an organic matter-rich mud. And that's where you form pyrite. And these trace elements that were once taken up by these organisms are released and then in turn absorbed by sedimentary pyrite. And that's what we measure. So it's essentially, we have a measure of what the trace element of the water column may have been like when the pyrite formed. And when you look at that over time, it gives you an idea about how the water column trace element chemistry has changed through time. And that's essentially what we want to track. So this is an example of what an outcrop looks like, a typical black shale, marine black shale. And if there's a close-up of the pyrites here, which is what we love analyzing using the LA-ICP-MS. But outcrop samples aren't that ideal for my study. I prefer drill cores because the pyrite isn't oxidized. I can't tell you how many drill cores I've logged over the last 7 years, probably thousands. This is what a typical drill core marine black shale looks like. You look for the pyrite, you cut the piece of rock accordingly, and then you make mounts out of it. In this case, I'm showing you a pyritized ammonite, and this is a non-pyritized ammonite. But essentially, this is what a laser mount looks like. And before we use the laser ICP-MS to analyze our pyrite, one key critical step is to look at it under the microscope. Anyone who's looked at a pyrite or a black shale laser mount under a reflected light, you'll be able to tell the various different types of textures that you see. And it's very important to, at a very early stage of the project, to be able to characterize the different textures of pyrites, because they mean different things. And so I think petrography is a very key aspect of essentially any research, but also very important for our project as well. So pyrite types, you get very coarse hydrothermal pyrites, and then you've got the beautiful raspberry framboidal pyrite. That's what I like, but not all sedimentary pyrite are sensu stricto framboidal. They can be various types of sedimentary diagenetic pyrite. And here's an example. And I'm not showing you the data here, but these photographs are essentially taken from the same laser mount. So just to show you the variety of pyrites that you may find in your rock, but they are useful for different purposes. For the purpose of ocean chemistry, we would like to stick to sedimentary pyrites only, of course, for obvious reasons. Well, once we've characterized the different pyrite textures, we use the laser ICP-MS to analyze a suite of trace elements in pyrite. And then, of course, we're now using in-house ladder software, which is how we process our data that we gain out of the laser machine. So as mentioned earlier, I've got two main research aspects. One is for exploration and the other for ocean atmosphere redox evolution. I'll start with that first and then tell you a bit more about the exploration side of things later. So I think my title would have given it to you, well, Brilliant Billion. One day she's talking about the boring billion. And if some of you've read Alan's recent press article, and he said the boring billion is a very catchy term. And it's funny how only geologists would find the boring billion a catchy. No wonder people from other sciences think we're weird. And I'm guilty as well. It's a catchy term. The only problem is that it's not a boring billion. But I'll give you some insight as to why it was called the boring billion. I think people are coming around now and not accepting it anymore. But when I started my PhD, it was very much, very popular. Oh, this period is the boring billion in Earth's history. So this is, I'm sure everyone's well-versed with Earth's timeline. So the period from 1.8 to 0.8 billion years is popularly or infamously known as the boring billion. And let's have a look at a very, very simplistic schematic representation of evolution of life on Earth. So we basically started from being very small and microscopic in nature, and then things got more and more complex. That's how we like to look at it. And well, this is probably the benchmark of complexity, I suppose, Freddie Mercury. But essentially, yes, life started out as being really simple and microscopic to more complex to present days. So what would this look like if we were to look at it in the Earth's timeline like this? So right from microscopic metazoans or animals, in other words, to very complex life, all occurred during this period here in the last 500, 600 million years. Prior to that, life was very microscopic and simple, as many would like to refer it to as. And the term boring billion came about, well, I think it was proposed by Roger Buick in 1995. And he referred to it as the dullest period in Earth's history. But the boring billion term was coined by the late paleontologist, Professor Martin Brazier, where he looked at carbon isotope signatures in carbonates from the MacArthur Basin and several other sedimentary basins. And he concluded there wasn't much fluctuation between 1.8 and 0.8. And he attributed that to some sort of biological stasis, because carbon isotope signatures are intimately tied with the biological cycle. And so he assumed that that period may represent a period of biological stasis. And yes, the boring billion term just absolutely flew from there. You started seeing articles. It was very depressing for me because 2014 was when I started my PhD. There were articles like, why did evolution stall during the boring billion, poison-filled boring billion years, fluctuations stalled life on Earth, boring billion years blamed on sulfur-loving microbes, billion years of starvation, billion years. Most boring time on Earth. That was bad, yeah. Why didn't animals thrive on Earth sooner? Again, it's a boring billion to be blamed. And this is my favorite. Life on Earth was nothing but slime for a boring billion years. And guess who published that? My own supervisor. So yes, it was quite something to begin my PhD research with this sort of literature review. But I'm really glad to see that things have changed. I mean, these are articles from March 2021, couldn't get any more recent. And I So this is the article I was talking to you about, where Alan Collins suggested that we perhaps should follow the lead of geologists Peter Kaywood and Chris Hawksworth and call it the Middle Age. No more the boring billion. And now it was very pleasing to see this, really. So why did people start calling it the boring billion? I think that is potentially because of the fact of this really strong emphasis on oxygen in evolution of life. And I really think it's a very human way of looking at evolution of life, because oxygen is a major part of our sustenance. And so we look at everything, and particularly for macroscopic life forms, as oxygen being the most important driver for evolution of life. And this is a very, very famous curve. Everyone in my field would use it. It's from Lyons et al. 2014, about how oxygen evolved through time. And it's really this uncertainty band here, because you don't know if it was this high or whether it was this low. This uncertainty here is what really researchers use as a way to explain why oxygen may have been in very low concentrations, and perhaps that's why it stalled life. But then you've got people like us. This is a paper by Large et al., our group here, and we think that uncertainty band is not as simple as it looks in the graph. It could be more dynamic, as shown here. So really, there's a lot of room for research, lots and lots of evidence to suggest everything was quite static. Lots of evidence also suggests that it was very dynamic. It depends on what geochemical proxy you're using. But regardless, I think a multi-proxy approach these days will probably provide more insights into how the oxygen varied. And the other thing people love to emphasize, other than oxygen, is everything was small and microscopic, so it's obviously not important. And this is a snapshot of when I was at the Harvard Museum for Goldschmidt conference. And I was intrigued because this shows, this is what this big circle here is, the microbial diversity, and this is animal and plant diversity, the single dot. So even though they're small and microscopic, there are probably more microbes on your fingers right now than there are people in India and China. Just the sheer vastness of microbial diversity need not be underestimated whatsoever, just because you cannot see it with the naked eye. So a lot of emphasis on oxygen and a lot of emphasis on macroscopic organisms. And because you don't find either of those two things, oh, well, it must be boring. But like I said, as you see more research, you realize oxygen wasn't required in that high levels by primitive organisms anyway. And that's been beautifully demonstrated by Mills et al. 2014 and De Navarro 2010, where they were the first to demonstrate that oxygen in some instances was not required at all. And in some instances, it was required at very minute levels. So then that brings to the question, and it's not something we didn't know. Even today, you and I can survive with oxygen for sure, but not just oxygen. You'll obviously need other elements as well. And that's where I think the whole pyrite work fit in very beautifully, because we were not only reflecting on how oxygen changed through time, but also other nutrient elements that are essential for life. So what are elements? Other elements are essential for life. Oxygen, of course, not for everybody, every organism, I mean, but it is essential. Then you've got your macro elements, and then you've got trace elements. And using the proxy, the geochemical technique that I use, I'm only able to understand the variability of the various trace elements in the oceans through time. And they are quite significant. As you can see, even Superman needs it. But in general, trace elements are incredibly important for basic cellular functions, as I mentioned here, growth and cell division. But they also form an important part of enzymes, not just giving them structural complexity, but also various different functions depending on what trace element is present in the enzymes. And you really need these enzymes, as we know, are catalysts, because the temperature and pressure where life can sustain are not conducive enough for the reactions that sustain life. And it's a complex chain of reaction, and we call it metabolic pathways. And it's a complex chain of reactions. Those reactions cannot take place. It's not chemically favorable under temperature and pressure conditions that are conducive to sustain life. So you've got to somehow speed up the reactions, and that's when you need enzymes and trace elements factor in there. And when I say metabolic pathways, as you can see, it's very easy and simplistic. No, the only aim of showing this slide to you is just to acknowledge how complex these metabolic reactions are to sustain life, and they need to be occurring at a much faster rate. And enzymes and the trace elements that form parts of the enzymes are absolutely key towards regulating these metabolic pathways. So they're incredibly important. So just emphasizing on oxygen through time just doesn't seem right. I think we need to look at a suite of elements, a suite of bioessential elements, and how that collectively have evolved through time. And also to say something is microscopic and not important is also quite naive, because when you look at some of the milestones in early evolution, without which there would be no microscopic evolution, here's what we've got: the prokaryote to eukaryote transition. So a transition from a very simple cell, such as that of a bacteria, to a very complex cell that you and I are made up of, 34 trillion of those we're made up of, occurred much earlier in Earth's history. Multicellularity, you and I are multicellular organisms, and origin of sex for facilitating diversification. So these were some really key milestones in early evolution. And I'd like to emphasize again that even though it does not manifest itself as a gigantic fossil of a dinosaur, which might pique our interest a lot more than a tiny microfossil somewhere in a black shale in the Proterozoic era, they do represent very significant biological events that have occurred in Earth's history, without which subsequent macroevolution would not have occurred. So it's important for us to acknowledge these milestones in early evolution. And it's not just fossils. You've got, earlier the data was of 540 million years is when evolution took place. But as we're getting, advent of new technologies, as I mentioned before, it allows you to see through or zoom in a bit more closely than we were previously able to. And there's the body of data just continues to grow and provides us more information, which pushes the date further back in time when we thought it wasn't possible. So this is a paper 2017 that I was very pleased to see about talking about the first plant microfossils in the boring billion. Then you've got fossils on one hand, then you've got traces of fossils. Again, there are talks of trace fossils in 2.1 billion years ago. Again, this is new data that has come out. I was very fortunate to go to China for some fieldwork before COVID-19. And I collected some samples. I've not published them yet, but I collected some rocks from the Mesoproterozoic successions here that are equivalent to the MacArthur Basin in Northern Australia. So I wanted to compare how the chemistry worked, but I also bumped into these really interesting, which I think are trace fossils, but I'm continuing to work on them. So this is just a cross section of this one here. And yeah, I found these intricate structures on the carbonates that was underlying the black shell that I was interested in sampling. So you can find all sorts of things if you look closer. And so that was the fossil side of things. If you look at molecular clocks, that also suggests that, it can be pushed further back in time into the Proterozoic when people thought it was boring. So you've got I gave you a little summary of all the information that we have now of the Proterozoic that we didn't have before. Just to summarize it, this is what we have. And it makes sense to have these stepwise evolutionary milestones achieved first before you have a macro evolution in Earth's timeline. So these were very critical events that have taken place And you've got fossil evidence, molecular clock evidence, you name it. And it's a growing body of data. Earlier on, this was all blank and people assumed it was boring billion, but obviously not anymore. So yeah, my research really now tries to understand what this has to do with the chemistry of the oceans that I understand using pyrite. Oh yes, there's another, would have called it a fun fact normally, but considering we're in the middle of a global pandemic, I've crossed out the fun. The machinery that the viruses use to break into host cells, I'm sure you're all familiar with that now. Also, for some reason, the evolutionary journey seems to have come about around 1.5 billion years as well. I'm not sure if I'm proud of that, but I just thought I'd mention it. So like I said, when, how, and why. Why is where we feature. Why things panned out the way it did is where geobiologists and geochemists such as myself would throw an insight. But when and how is something that I cannot talk about. This is not my expertise, it's biology. And I think something I've come to realize in the last 7 years is looking at evolution of life. It's an extremely dynamic topic. And as geologists, you can only play your part. And I think my part is the why. But it's important to acknowledge that there are questions as to when something happened and how something happened. When I say something, I mean evolutionary breakthroughs. So this is again a schematic representation of a eukaryotic cell that you and I are made up of as well, and all complex life on Earth is. So when and in what order were these cell components engulfed into this bigger area here is intriguing, and it's fascinating, and that's something biologists are trying or having, or I don't know if they've completely have, but they are trying to figure it out and continue to do so even now. When and in what order? How, we know something about how. It was either the Endosymban Theory that was proposed by Lynn Margulis, where two simple cells came into a mutual symbiotic relationship where one benefits from another. And you see examples of that in today's biotic realm as well. Maybe it wasn't that lovey-dovey and it was predation instead, that one larger cell engulfed the other not so larger cell and lived in a sort of a mutual relationship, but one phagocytosed or predated on the other one. And that's how the various cell components were concentrated within a larger area, like I showed in this slide here. So yeah, whether it was endosymbiosis or whether it was more drastic and aggressive like predation, again, examples of which you can see today, is a question mark. We don't know. But you've got evidence for both of those in the early rock record. And these are process-bearing microfossils. They're not hypothesis. You can actually see them under the microscope. So you've got evidence of how, but it's very hard to pinpoint exactly how it happened. But there are various theories. Now, why is where I think us geochemists fit in. And that's where we can say deep time tectonic setting. What were the plate movements like? Were we in an active tectonic zones? Because then erosion rates would be higher. And if erosion rates are higher, combine that with higher atmospheric oxidative weathering, then you get a higher nutrient flux into the ocean. So a combination of these geological processes have a huge significance on the nutrient input into the ocean and availability of the various elements for life in the oceans. So the why is where we probably, as geologists and geochemists, we feature. So oxygen, important macro elements, the trace elements is what basically we're after. And as I mentioned, I have been looking at the concentration of these trace elements, colored in red here, in sedimentary pyrite in marine black shales through time. And in this slide here, I'm basically showing you the variability of these elements here, which I've selected because they're very bioessential for all life forms. And I'm trying to look at its variability from 2000 to about 500 million years ago. So what you can see here, and this work was published in Nature's Scientific Reports. This is one of the major outcomes of my PhD. And basically what we tried to show was that at the start of the boring billion, there was a decline of nutrients. And we're specifically talking about these elements here. And that then about at 1400, you see it go up again above this critical level here. And so we have a period of low trace element availability followed by increase in trace elements. So how does that compare with all the evolutionary events that I've been talking to you about? Well, all the biological innovations, the endosymbiosis or phagocytosis, whichever way you want to explain how the first complex cell formed, the fact is it did form in this time here. So whether two cells came into a mutual symbiotic relationship or whether one cell ate the other one, regardless, they came together and formed the first complex cell. And there were other biological innovations such as multicellularity and the advent of sexual reproduction. And then following from 1400, you see a diversification of eukaryotes where you see microfossils of eukaryotes indicating that they've spread their habitats and they've diversified. So how do you then explain that? And the way we did it was that this period of low trace element probably created some stress because lack of nutrients is when you would think that organisms would be pressured into looking for alternative mechanisms to sustain. Of course, there would be casualties. There's no question about that. And I think to give you an example, it's even now with the lockdowns and everything, obviously some businesses went flat, but then it also prompted other businesses to look for creative strategies to cope with the situation. I think it's a similar situation in the sense that this was probably the first striking variability in nutrient concentrations on Earth. Now, it was probably due to oxidative weathering, plate tectonics, etc., whatever may be the reason, but it was low, and that would have coerced organisms to opt for elements that were available. They may have been used to zinc, but they now have to substitute that with cadmium. It was available owing to similar chemical properties. And once availability of elements change, their utilization strategies will also change. And that may have given them an opportunity to evolve because different elements will form different functional capacities in the enzymes that I was talking to you about, and they will have different functional properties. And so something that they couldn't do before, they probably can do it now. So it's that low period that potentially everyone would love to call it the boring billion. I think that's the most significant part of the boring billion because the organisms, probably experienced the first ever striking variability in nutrient concentrations, and they had to come up with adaptive strategies to sustain life, and in the process evolved. And when conditions got better, then of course diversification and radiation of eukaryotes occurred. So in really short, no pain, no gain is what I'm trying to say. And that's how we described the nutrient fluctuation through the boring billion. And needless to say, it wasn't boring billion at all. I've never believed that. Taking that study on in my postdoc, we've added more data to the database. This is just one way of looking at it, a correlation matrix, which gives you an idea what elements were high, what elements were low. Again, I'm not trying to confuse you by showing this slide. All I want you to acknowledge is that it's not one element. It's a suite of elements that change. So the elements are here, and they're all in different concentrations. So just because one element is low or if oxygen is low doesn't mean the whole thing has to go down. Organisms, particularly microorganisms, are incredibly smart, and they are far more resistant to changes than we are. And so they can cope with these stressful conditions a lot better than I suppose we can. But really, the objective of showing this slide to you is just we can collect more data. And I think with more data, this correlation matrix may be modified or revised, but it's giving you some idea. There's not just one, but co-evolution of several nutrient trace elements, and that will have affected evolution of life, because that then dictates what element is available and what's unavailable. And that then affects the trace element utilization strategies by organisms and then their functions and evolution. So we published that in Geology last year. So yeah, I'd love to know your thoughts if you ever bump into this. We called it the pyrite edition because there's a lot of work being done on bulk, conventional bulk rock, but very little large-scale database on in situ analyses, such as ours on pyrite. So yeah, we published that. Just very quickly on the utilization of pyrite chemistry to exploration. This is just a snapshot of one of the earliest zinc production sites in the world, probably. I've actually been there for my fieldwork. It's a beautiful place in India. So why zinc? Well, it's incredibly important. It was very important in the past. As you can see, zinc manufacture moved to China in the 17th century, and 1743 is when Europe saw its first zinc smelter. So it was a key important commodity in the past. Andrew Simons obviously needs it for when he's playing test cricket. Apollo 11 utilized it as well for its hearing aids, which was one of the very good spin-offs of the Apollo 11 mission, and of course for galvanizing and various antiseptic ointments and sunscreens, of course. In the future, it's definitely part of one of the green metals. So we can have this guy take the burden off him and make sure they have a better life. And so, yeah, in order to lessen that gap, we need zinc as well. And so one key aspect, because I was looking at so many black shales, there were black shales that host some of the world's largest SEDEX zinc lead deposit. And MacArthur River deposit happens to be one of them, and it's hosted by the Barney Creek Formation. I wanted to see if mineralization affects the seawater chemistry in any way, and it does. So what I did was looked at samples that's furthest away from the ore body, intermediate and very close, and I wanted to see if there's any systematic change in trace element concentration as for when you are near the ore body and when you move further away. And if there is, then can industry use that information to look for more zinc deposits in the area? And as expected, there were elements that decreased as we moved towards the ore body, such as nickel and cobalt. There were elements of economic importance, obviously, would increase as we move towards the ore body. And there are elements that remain the same. So you can play around with this data, and I'm currently leading an industry project which looks just that, and we've got a huge body of data, and we hope to revise both of these. These were done in 2017 during the course of my PhD, but I'm currently working on adding, 5,000 more analyses to these two figures here to see how this will shape up. And this is very useful information just to give industry idea as to what an unmineralized black shale chemistry from 1650, that's how old the Barney Creek Formation is. And then as you basically move closer to an ore body, the chemistry changes. And so if that can be used for exploration vectors, and that would be fantastic. And that's something I'm working on very closely. So in conclusions, really, as I mentioned to you before, you can use trace elements in pyrite as an exploration tool, as I've just mentioned very briefly. And I had a very simplistic slide to show you furthest, intermediate, close. We're obviously working, on adding more to this database and the various nuances that come with it. But it's a very useful technique because it's in situ and it's extremely sensitive to any variation. So if you're around an ore body and the halos around it, it can really effectively track those halos around a deposit. Can be very, very useful. And the other important thing is to ascertain background chemistry, because that will minimize exploration risks as well. And the other, of course, modifying this curve as we have more data, look at other elements and apply more statistical treatment to the data to get a robust trend of nutrient variability through time in Earth's history and how that compares with available paleontological information. And yes, I'd like to emphasize that anything that's low or below critical point does not necessarily mean doom and gloom. And anything that's high doesn't necessarily mean, oh, lots of nutrients, it's good. It's like trace elements. With you and I, if we have too much of something, it's toxic. If we have too little of something, it's deficient. So it's not that straightforward, particularly with trace elements. So quantification is something that I'd really like to do. And then, of course, it'll vary with different organisms. Normally, I would conclude my slide with a thank you, but I will take this opportunity to talk about very briefly about something I'm extremely passionate about, and that's the status of women in India and in general, of course. But I am from India, and I keep track of things, how it's going, and it really sometimes disappoints me greatly. A recent report, the Global Gender Gap Report, demonstrated that we are the third worst performer country in South Asia in World Economic Forum Global Gender Gap Report. And yes, the pandemic really did hit us hard, and particularly women. Like I mentioned here, 120 million people lost their jobs, but they were mostly women and girls. Whilst the men have been able to regain most of their jobs, women still haven't. Much of the cultural economic progress made by women seems to be going in a backward trend because of the pandemic. And another startling statement is that even though we have 48% of our population as women, we only are contributing 18% to our country's economic output, solely because of the lack of opportunities available to women in India. And yeah, I'd just like to show you the picture of two women who feature and lead projects in India's, rather I should say, one of the world's leading space research organization. Highly intelligent women contributing in a major way to these space research programs. We have women who are working really hard on the ground in terms of skilled labor, India being an agricultural country as well. We also have women who champion various different causes, particularly LGBTQI plus communities in India, which is a very touchy topic, and it's sometimes very dangerous, and yet women are out there championing the causes. And we have women such as these who are differently abled but still represent the country in the cricket disabled nationals team. So just modifying what Helen Reddy said, I'm a woman, hear me roar, but really what I can hear these women saying, I'm a woman and I want more, and they deserve it. I'll leave it at that. Thank you.
Thank you so much, Indrani. That was a wonderful talk, and thanks so much for bringing, this to our attention, about gender equity in India. It's not something we hear about in our news cycle at all. So it's really important for people to have an increased awareness of what things are like in other parts of the world. And those women are incredibly inspiring, and I definitely want to know more about them, and I'm going to look them up. So thank you for that. We have quite a few questions coming through on the chat already. So I will just go straight to that. And if anyone has any other questions, you can pop them in the chat or unmute yourselves and yell out when you feel the time is right. Okay.
Sorry, Melanie, I just thought I'd also like to thank Hannah Moore, whose office I'm currently using. I meant to, just say thanks to her because I forced her into a temporary eviction during the course of this talk. So thanks, Hannah.
You're very welcome. Fantastic. Okay, we have a question from Joanna Parr. She says, great talk, Indrani, fascinating. Have you considered what happens to the trace elements in pyrite as it is metamorphosed? With fairly low temperature metamorphism, are we losing any of the primary trace element signature?
Yes, it's an excellent question. So in the first paper that we published was the proof of concept paper, Joe, and we demonstrated, I think it was from Sukhoi Log, where you have beautiful framboids and then you've got really coarse grained pyrites in the same, and the coarser grained ones were metamorphosed. And most of the trace elements, yeah, they were exolved out because the pyrite was more pure pyrite with less trace elements in there. So we don't use pyrites. It's best if we don't use metamorphic pyrite at all. But from my research, I think we used upper green schist facies in Zawa, which is 1700 million years. And I think green schist facies is about when things start to get, you can't take it seriously. But I think until upper green schist facies, you should be okay. But I'm very paranoid about using metamorphic pyrites altogether, because it comes up over and over again, and we've tested it in various different time period for different pyrites and metamorphic grade, and it's usually very low. So yeah, it's probably not worth using a metamorphic pyrite in this case. But upper green schist facies is whereabout you can use if you're desperate.
It strikes me as it would be a good study to actually correlate the level of metamorphism with the mobility of the trace elements, and that's not only for pyrites, for a lot of minerals.
Yeah,
that's great. Thank you.
Thanks, Joe.
Thank you. Okay, we have a question from Hector. He said, fascinating talk, thank you. About trace elements, is there some kind of consensus on a hypothetical path of the order in which each became incorporated into life? And when you refer to this period of scarcity of trace elements inside the boring billion, I guess it's established that all of them were already necessary to sustain life? Or would it be possible that one or two may have been incorporated for the first time precisely in that period of trace element stress?
Yeah, so that's exactly what we talk about in our geology paper. So coming back to your first question, what order were the trace elements being utilized by organisms is very hard to, very hard to pinpoint. It's rather impossible, I should suggest, unless we've got some crazy analytical technique that might come in the next few years. But I think you could still predict it based on what was available in the ocean. So through plate tectonics, you know what basically the source rocks that were available for erosion and weathering. You can keep track of that. And if you know that, then if you know what your source rocks were, then you'd probably be able to guess what suite of trace elements went into the oceans, whether it was the trace elements from mafic rocks or whether it was felsic rocks. Again, this is very general. But yes, what order the trace elements were being utilized by organisms would be something incredibly hard to pinpoint. But we did have, so I'm involved with a group in New Zealand, and they wanted to basically test this stress hypothesis that we proposed, but again, on modern organisms. So even if we do see how organisms take up various elements and in what order today, don't know if you can extrapolate that to the past, but it's a great question. I would love to know that myself, about how, in what order. But like I said, you can still comment on what was being made available in the geochemical environment for organisms at various points in Earth's history. Again, very first order. Everything I talk to you today is very, very first order. Obviously, there are, it's very dynamic. And if you keep zooming in, you see second, third, fourth order changes. But on a larger scale, I think you should be able to comment on a suite of trace elements. But exactly what element went in what order, I think it's very hard. Mm-hmm. Was there another question?
Well, we might move on because we're running a little bit out of time anyway, and there are quite a few more questions in the chat, and we'll try and touch on most of them. So we've got a question from Ian. Is the low and high availability of trace elements a global event, or is it a local ocean event? And he also asks, are similar studies being undertaken on pyrite and coal to reflect on freshwater paleo environments?
Yeah, so this local versus global is something, you sound just like my reviewer there, but it's something that we really like to think about. And I think for the boring billion, I can safely assume that the conditions, and it's not just pyrite chemistry, but you look at the tectonics and the supercontinental reconstruction, the Yan Liao Basin and the MacArthur Basin, as well as rocks in the Vindhyan, so the Proterozoics in India. And you see the Proterozoic in America, United States of America, and the Rocky Cape group in Tasmania, and there are parts in Russia. So you see the same paleontological information come out of these areas. So similar eukaryotic fossils that underpin the paleontological information. Chemistry-wise, the Yan Liao and the MacArthur Basin, same age, they're very similar. And also the samples from the Vindhyan supergroup. So yes, we don't have the same age rocks from different geographic locations for all time periods, but for the middle Proterozoic, when I've looked at India, China, United States, and obviously Australia, they are very similar, not just in terms of the paleontological information, but also the chemistry of pyrite. But yeah, it would be worthwhile trying to see, and we're working on that actually, to have same age rocks, but from different parts of the world, and comparing and contrasting as to how the chemistry changes.
Well, that sounds really interesting. And we'll leave this our final question. There are a few nice comments as well in the chat that you might want to read. But the final question we'll have from Owen. It says, the trace elements which are essential for life are an interesting bunch. I'd never considered their links to pyrite before. You discussed cadmium as one element, for instance. Is it essential for any life? I would have guessed that cadmium was more inhibitory, but as you said, the boundary between essential, trace, and toxic is very thin sometimes.
No, cadmium is essential, and it is, and so that's something that's evolutionary conserved. So a lot of the prokaryotes use zinc, but because cadmium is very similar to zinc, when zinc is unavailable, cadmium may be utilized. And there's a very good paper by Morel et al. I think it's the 2010 paper, where he talks about various different elements, but he specifically points to the fact that usually you just have organisms using zinc, but at one point when zinc was not available, cadmium may be utilized. But again, it's like with anything, yeah, you can't have too much of everything or too little of anything, but cadmium can definitely be utilized.
Excellent. Okay, thank you so much, everybody, for attending the WMISA seminar today. And thank you so much, Indrani, for a wonderful, wonderful talk. I really enjoyed it and I learned a lot, as I'm sure many others did as well. So with that, we'll draw to a close. Thank you, everyone.
Thank you.